Thank you for viewing this Industry and Science on Demand session. If you need assistance at any point, please request support located along the top of your screen, indicated by the question mark. Please enter questions or comments in the live chat section. Speakers will be monitoring the chat and will respond to questions. You may also contact speakers through networking features on this digital platform. The speaker directory is located on the dashboard. Participants are also encouraged to visit the speakers for more discussion time in their booth located in the exhibit hall throughout the meeting. Enjoy the session.
Good morning, good afternoon, and good evening, wherever in the world you are tuning in from. Welcome to the scientific session on culture-independent rapid diagnostics, impact on sepsis management. I am Dr. Aparna Ahuja. I'm a strong advocate for patient and healthcare worker safety with a career of almost 3 decades in laboratory medicine and medical affairs in medical device in vitro diagnostics. Currently, I'm the Chief Medical Officer at the T2 Biosystems and serve as the company's key medical spokesperson, providing leadership and strategic direction for all medical, clinical, scientific, and regulatory activities of the company. I am Corporate Advisory Board member for American Association of Clinical Chemistry, Chair of the New Jersey section, CLSI Expert Panel member, and auditor with College of American Pathologists, ISO 15189, and also an active member of various laboratory and medical associations, such as American Society of Microbiology, College of American Pathologists, and AMP. I would like to welcome our speakers, Dr. Jim Snyder. He is going to be talking on implementation of T2 Candida and T2 bacteria panel. He is a professor of pathology and laboratory medicines and serves as the director of microbiology and infectious diseases molecular diagnostics at the University of Louisville Hospital. He's board certified both in medical and public health microbiology and is a fellow in the American Academy of Microbiology. He holds professional memberships in the American Society of Microbiology, Pan American Virology Society, Infectious Disease Society of America, and the South Central Association for Clinical Microbiology, for which he currently serves as the president. He recently served as the coordinator of the Sentinel-level laboratory protocols for biothreat and emerging infectious diseases as part of the Laboratory Response Network. He serves as a member of the American Society of Microbiology Expert Panel for the development of evidence-based laboratory guidelines. I'd also like to welcome and introduce Dr. Todd McCarty, who will be speaking today on the impact of T2 Candida utilization on clinical outcomes. Dr. McCarty is board certified in both internal medicine and infectious diseases. He is the Fellowship Program Director for Infectious Diseases at University of Alabama at Birmingham Hospital. He's also the coordinator of antimicrobial stewardship at the Birmingham VA Medical Center and a member of Antimicrobial Stewardship Committee at UAB. He serves as the principal investigator on multiple clinical trials evaluating new antifungal as well as antibacterial agents. Please note that we will have a panel discussion at the end of the session, and also the speakers will be present at the T2 virtual booth on June 21st at 1:30 to 2 p.m. EST to answer any follow-up questions that you have after the symposium. Sepsis is a burdensome condition worldwide in terms of morbidity, mortality, and healthcare costs. Sepsis represents $62 billion in US healthcare costs, claims more lives each year than 3 leading causes of cancer deaths combined, the lung, colorectal, and pancreatic. CDC data states that each year 1.7 million adults in the US develop sepsis, 270,000 Americans die as a result of sepsis, and 1 in 3 patients who die in hospital has sepsis. Another concerning fact is that hospitalized COVID-19 patients face a 22% higher risk of developing sepsis and are at 113% more likely to experience septic shock compared to hospitalized influenza patients. There has been a global rise in antimicrobial resistance infections, which has resulted in increased morbidity and mortality for patients of all ages and background. And there's another fact that antibiotic resistance disproportionately impacts the most vulnerable, and it's more seen in low and middle income countries. The CDC estimates that more than 2.8 million antibiotic-resistant infections occur annually in the US itself, accounting for more than 35,000 deaths. Prompt administration of antimicrobials with activity against the causative pathogen is the cornerstone of sepsis management. Indeed, delayed administration of optimized antibiotics is associated with progression from severe sepsis to septic shock and increased rates of adverse events, mortality, and healthcare costs. To reiterate, extended time from presentation to appropriate therapy remains a major contributor to poor patient outcomes and proliferation of antimicrobial resistance. Current standard of care requires up to 3 days to provide sepsis ID and susceptibility results, which can delay time-sensitive, life-saving treatment. And for every hour delay in time to appropriate therapy, survival decreases by 7.6% during septic shock. Thus, every hour is so important. As many as 80% of sepsis deaths could be prevented with rapid diagnosis and treatment. One of the key risk factors associated with development and spread of antimicrobial resistant organisms is the prolonged use of broad-spectrum antimicrobials. Thus, the time to appropriate therapy is a key driver of clinical outcomes. So as we understand that the time to therapy is very critical, very important, so why wait for results? T2 Biosystems diagnostic panels are the only CE-marked and FDA-cleared tests that provide species identification directly from whole blood within 3 to 5 hours of the first blood draw. So if you see this, from the blood drawn from the patient till the positive blood culture, the time taken could be between 1 to 5 days. And while there are many rapid diagnostic tests, the culture-based rapid diagnostic test can provide species identification quickly within 1 to 3 hours, but that can be done only after a positive blood culture. Thus, they cannot be leveraged until the culture becomes positive, which takes days. And also they cannot be utilized in cases of culture-negative sepsis. Culture-negative sepsis is a common clinical concern where patient appears septic with a lack of pathogen growth on culture, which could be due to many reasons. And one of the common ones is a prior intake of antibiotic. Then the non-culture based tests, which are not reliant on culture positivity and thus are not influenced by interference like antibiotic intake prior to the test, can be done very quickly directly from blood. And T2 is one of those, and maybe the only one that's available right now. Coming to the two panels that would be discussed in the session today, the T2 Candida and T2 bacteria. They are run on a fully automated T2 DX instrument that you can see in this picture. And the result comes in 3 to 5 hours. There's no sample preparation. Sample needs to be collected in an EDTA tube, a whole blood sample, which can be inverted and put into the panel and into the instrument. And it's very sensitive with an LOD of 1 CFU per mil. So both the Candida and bacteria panel are highly sensitive, more than 90% is the sensitivity of both, if you see in general, and specificity is also very high, 98 to 99.4%. Both are FDA cleared and CE marked with a very low CFU per ml limit of detection. The T2Dx instrument workflow, as you can see here, as I already mentioned, you need to have a 4 ml patient blood drawn in an EDTA tube, which is then loaded into the instrument and onto the T2 diagnostic instrument. There, within minutes, the blood cell is lysed, concentration and lysis of pathogen cells happen, and amplification of the target DNA and cluster nanoparticles, which are detected by T2MR. And the clustering is detected as a change in T2 relaxation time of hydrogen nuclei in the sample. This is a proprietary methodology enabling inhibition-free DNA amplification in complex clinical matrices. There's no background interference, example, either from human DNA or antibiotics, simplifies the process and eliminates any extraction and purification of target steps. T2 magnetic resonance, which we say T2MR, enables measurement direct from patient sample, which enables this higher sensitivity, which we discussed earlier. There is enough data, and it's a growing number of real-world T2 Candida and T2 bacteria success stories coming out, which shows the impact on the patient care and the antimicrobial stewardship optimization. Not going to the details, however, just mentioning a couple. Henry Ford Health System, a study there demonstrated a $2.3 million saving in the annual hospital and reduced median ICU length stay by 7 days, overall stay by 4 days. And most negative patient had antifungal discontinued or de-escalated, saving a lot of dollars upfront. So the healthcare costs impact is also immense. Another study published from Lee Health mentioned that there's a median length of stay reduction of 7 days, and unnecessary antifungal therapy was avoided in more than 50% of patients. Average net antifungal saving of $195 for every patient tested. There are similar stories in GMLA. T2 detected 20 positive cases missed by blood culture, and MALDI and BioFire. And T2 bacteria positive, while blood culture negative, results significantly more likely in patients receiving antibiotics. 66.7% of infected patients missed by blood culture and detected by T2 were being inappropriately treated at the time of T2 bacteria result. And Oshner, a study there, mentions that detection of more pathogens occurred in 11 patients where blood culture remained negative. 70% of patients with a positive T2 result could have experienced at least some clinical benefit from the T2 bacteria result. And T2 bacteria assay could have reduced time to effective therapy by an average of 28 hours. I would like to mention another study which has just been published last month. It is a meta-analysis of 14 control trials evaluating antimicrobial and resource utilization with T2 magnetic resonance for rapid diagnosis of bloodstream infection. And there were 7 outcomes which were evaluated, and these are the result outcomes. The time to detection was 3 days faster with T2. Time to species identification was 3 days faster with T2. Patient testing positive on T2MR received targeted antimicrobial therapy 42 hours faster, while patient testing negative on T2MR de-escalated from empirical therapy 7 hours faster. Length of ICU stay, 5 days shorter with T2, while length of hospital stay was again almost 5 days shorter with T2. Mortality rates were comparable between T2MR and blood culture. So here's where I'll end my little introduction and background on sepsis and T2 panels. And I'll hand over to Dr. Jim Snyder. Thank you.
Well, thank you. And I want to also thank T2 Biosystems for the invitation to speak in this session. I am fortunate to be teamed with the infectious disease specialist, Dr. McCarty, and I'm going to be talking from the analytical, technical end. Dr. McCarty will be focusing on what I consider very important, the clinical application and their experience. I also have to admit that I'm somewhat of the new kid on the block, although we've been working with this system off and on for the past 5 to 6 years. And I think it's important to understand some of the obstacles that we have been placed in front of us from the perspective that up until about 1.5 years ago, our institution was managed by a private hospital management team. And as a result of that, had very little understanding or appreciation of what an academic medical center is. We are a university-based academic medical center, trauma level 1 center for our region. As I said, we've been managed privately up until about 1.5 years ago for about 7 years. And I had become familiar with the T2 at least 6 years ago and became very interested in the Canada application, which at that time was the only application that was available. It was interesting, I got a call from the original vice president of operations of this management company. He said, I want to call your attention to new technologies out there that will completely replace blood cultures. And after he mentioned the T2, I said, I beg to differ. It will not replace blood cultures. You still need the isolate for, if necessary, confirmatory identification. And most importantly, in this case, antifungal susceptibility testing. And so I was successful in convincing him to endorse a study. Well, during that period of time, the contract with that agency was terminated, and we were only able to do maybe 5 months worth of the study. So I will admit up front that our database is limited. So it's one of our limitations with our experience in looking at the T2 Canada. But at the same time, the bacterial panel became available, and I'll share with you our experiences on both of those. I have nothing to claim as far as conflict of interest as it relates to this presentation. So the objectives that I've selected for this presentation are listed there for you on the slide, but basically we are in an era of rapid diagnostics, and that concept is driving our laboratory into the future. And T2 is just part of that overall objective, with the idea of adding it to our service menu, along with several other platforms that fulfill the objective of rapid diagnostics. Physicians know it's available, they're asking for it, and I think we as laboratorians are obligated to make it happen if at all possible. We also know our responsibility to support antimicrobial stewardship, and we found that possibly with the T2, both the bacterial and the Canada, that would allow us to support antimicrobial stewardship. This is also a multidisciplinary process. So over the period of time that we performed the analytical assessment of the technology, our pharmacy colleagues, and I should also indicate that we have 2 facilities in addition to our university hospital, of which we serve as the core laboratory. So 2 hospitals, the university and 1 other hospital in our network, were associated with this initial 5-month review of the performance and potentially the impact of the data on patient management, therapy, and et cetera. So it's a multidisciplinary process, and I'll say more about that as we continue. And I'll try to present a case or two to show you examples of how this is impacted. Now, those cases are more associated with the Candida than it is the bacterial. The bacterial is the most recent evaluation we have completed. We have not done any outcome studies or anything associated with it, but we do have data for the bacterial. So with that, Candemia, as you know, as once again summarized in this slide, is a serious problem. Prevalence ranges anywhere from 2 to 11%. In our respective institution, it ranges around 5%. It did creep up to 7% a couple of years ago when we added the other institution to our core laboratory. But be as it may, mortality does reach or approximate nearly 40%, even in the presence of antifungal therapy. So if you can get them on antifungal therapy much sooner, that should be able to reduce or hopefully reduce mortality. We also know that blood cultures lack sensitivity, around 50% sensitivity for confirming or even probable cases of dissemination as it relates to candidiasis. We also know that invasive candidiasis, it's not uncommon to have negative blood cultures. And of course, we know that culture takes anywhere from 2 to 3 days. The exception being Candida glabrata, which may take up to 4 to 5 days, but on the average, 2 to 3 days to get a confirmed culture. And the bacteremia side of it shares similar problems as those of fungemia. So we looked at the overall situation and said, how best can we use such technology? Should it be open-ended or should it be restricted to certain patient groups? And given the prevalence of, in this case, Candida infections, along with bacteremia, but primarily Candida, we knew that the high-risk patients that we have listed there, the intensive care units, the transplants, both organ, solid organ, as well as bone marrow transplant, oncology, our emergency room, and infectious disease can or will be permitted to order the test as well. They will make the decision, and I will mention later the guidelines that we have sent out to promote the optimal ordering of the assay in our efforts to promote diagnostic stewardship, but ultimately antimicrobial stewardship. Obviously, we have to take in the big picture of training the people who will be collecting the blood, the phlebotomists, the nursing personnel, because it's very, very critical, as I will mention throughout, that the T2 sample and the blood culture be acquired simultaneously rather than separately. Obviously, we wanted to compare the performance of the T2 to our standard blood cultures, both fungal and bacterial. I should mention that we only use standard bacterial culture broth. We do not use a fungal blood culture, except in certain cases. It's more or less an afterthought when we're looking for histoplasma, which is relatively common in our part of the world. Then look at what our data says, and then work with our pharmacy, our PharmDs, regarding clinical impact. So let's begin with T2 Candida. Now, this is a rather busy slide, I will admit, but we've worked with our PharmDs to really select the patients or help us to better utilize cases from those respective units that I just mentioned. I should add to this that in the enrollment, such criteria as the patient having unexplained fever or leukocytosis or a clinical deterioration or an ICU stay of greater than 72 hours. They were also eligible if they had a peripheral central line or a central venous catheter. Also, if they were on broad spectrum antibiotics for greater than 48 hours and were demonstrating clinical deterioration. Exclusion, though, was based on the fact that the assay was not performed within 24 hours of initial blood culture, and more specifically, if the assay was not accompanied by a blood culture that was excluded, or the fungemia was caused by a Candida species or a non-Candida species rather, such as maybe a Cryptococcus or Rhodotorula, which we did encounter, or death or comfort care resulted before the testing was completed. So you can see here we only had 25 cases. Ultimately, 10 were included. I won't go into greater detail, but we were trying to be as specific as possible, knowing that this in essence was a pilot study. So the results are such that we had 68 total specimens. As I admitted at the very beginning, one of the limitations of our study is the small database, but I think it establishes a trend. So we had a total of 5 T2 positives and 4 cultures. Of those 5 T2s, 2 were Candida albicans / tropicalis or Candida parapsilosis, of which there were 3. You will notice we had 2 T2s that were positive, but 1 T2 was negative, but the culture was positive. That turned out to be a Candida albicans. And why this occurred, I don't think anyone knows. It could have been something dealing with interfering substances. The paper out of the Indiana University group, Bokamp and others, felt that that was not the case, the way they had their study designed. I really don't have an answer for that, but I think that what it boils down to is we have to accept the fact and all know that analytical tests are not always or consistently or will be 100% accurate. So that's one of the limitations of diagnostic testing. We have to accept those. These are situations in which we would do an investigation with the patient, look at outcome, and et cetera. As we said, 3 parapsilosis, all 3 were culture positive. And you'll notice we had 4 invalids. Those invalids occurred prior to T2 Biosystems updating their systems. And since that time, we have not had any invalid. If we have, they've been very sporadic, but we had 4 during this 5-month period that we were looking at it. So the 1 T2 negative and the culture positive for Candida albicans is a concern and does require further investigation. So we have some ideas as to why this happened, but we need to be aware that this can happen. It's no different than the sensitivity of a blood culture, as we mentioned earlier, 50% or less, especially in fungemia, and not much better in bacteremia. So what we have done in our training is to ensure that the blood culture and the T2 sample are drawn simultaneously rather than hours or a day apart to avoid, if you will, that potential intermittent candemia and associated with the collection times. And once again, the group at IU believes that this is unlikely, so it's a challenge to find the answer. Another variable that could influence the results that we always have to take into consideration are the inhibitory factors, of which I'm learning more about. But like I said, the invalids, I want to make it very clear, occurred before the panel was updated in response to reducing the number of invalids. This has not been a problem. Overall, the low prevalence of Candida fungemia, we have to take that into consideration in the overall evaluation. We have to realize there's always that potential for a T2 negative, culture positive. And yes, you know, costs have to be taken into consideration, which I will mention when we discuss the multidisciplinary approach to going forward and justifying and requesting a piece of equipment. This is some of the outcome data that was put together by our PharmD, Dr. Nick Cottrell. I think one thing that stood out was our time to therapy in days. You can see the patients with positive T2 results received therapy much sooner than in our control group 2.5 days later. That's significant in getting patients on the appropriate therapy and preferably the targeted therapy. Once again, a small database, but it does establish a trend. What I did was extrapolated this data from what I've been able to gather from the literature. And even though our numbers are small, we're really not off that far in terms of impact. Now, the length of stay, we accept the fact it's probably not going to have a significant impact because these patients have, especially our transplant patients, bone marrow and solid organ, have underlying disorders, and they're going to be remaining in the hospital longer than most. Intensive care patients, we noticed, although I don't have it here on the table, about 1.5 days less length of stay. If you add up hotel costs or per day in intensive care versus what it costs us to do the assay, there's no comparison. It's far more expensive. We were able to look at somewhat at the 30-day readmission, and you'll notice in the control group, we had 1 readmit versus none in the T2 assay. Mortality, none in the T2 assay, and control. So I look at this or view this as a trend and pretty much similar to extrapolating it with what we know in the literature. So this is one patient that I can mention as far as a specific patient. This was an oncology patient, presented inconsistently with persistent spiking fevers. They were receiving broad spectrum antibiotics. They had a port in place. All blood cultures had been negative. The fevers continued. We asked or recommended a T2. The plan, though, was to remove the port and pray that the offending organism had been treated. Did not happen. The T2 was positive for C. albicans. Tropicalis actually turned out to be an albicans. The port was removed. The antibiotics were discontinued. Patient was placed on the targeted antifungal therapy. The fever terminated, and the patient was eventually discharged, which I think is a good news story. Let's take a look at the bacterial. These are our evaluation results based upon 76 samples compared to standard blood cultures. And you will notice that the T2 detected 8 positive, and there were 8 positive additional T2s versus 8 negatives. And those 8 negatives by standard blood culture involved Pseudomonas aeruginosa. There were 3 E. coli, 1 Klebpneumo, and 1 E. faecium. In the T2 positive blood culture concordance data, you can see there were 6 Staph aureus, 1 Pseudomonas, and E. coli. I think the interesting thing is that in the T2 negative blood culture positive, which there were 7, you'll notice that all the organisms listed there are not in the T2 database. So that's not surprising to have come up with a Proteus mirabilis, a strep pneumo. Whether or not that epidermidis was clinically relevant or not, I went back and pulled the record earlier today, and it was 1 of 3 blood cultures. We defined that as a contaminant. And the luthiensis was also regarded as a contaminant, and there was no question about the diphtheroids. So I think overall, the T2 bacterial data is very good. And obviously, we had 53 concordant negative by both culture as well as T2. So I'm satisfied with the fact that the bacterial panel, which we just completed verification on, is acceptable. So the multidisciplinary process, I cannot overemphasize enough when you're going after or trying to acquire new technology. The approval process, we had to do our laboratory cost analysis, and I'm sure all of you have been through that. We needed to determine our direct cost per reportable, estimate our potential savings to the institution. And I will be the first to admit, I'm a believer that the laboratory should be regarded as a center of savings and not a cost center. The savings that we can generate for the entire institution is well worth the effort. And as I said, it may cost us a little more upfront, but the thousands, if not hundreds of thousands of dollars that we could potentially save the institution, I think should be the driving force. We verified again both the Canada as well as the bacterial panel, and that verification data will be submitted with, well, we've already submitted our request to management, along with appropriate representative literature, both literature that's not so good versus literature that is supportive, but we wanted to be fair and balanced and transparent. The other area was to work on our workflow. What impact will this have on our workflow? We made a plea 2 years ago to establish a dedicated 24/7 microbiology staff. And within the last 8 months, that's been officially approved. So we are now 24/7. And as I will mention in our training process, those people that are working after hours will be trained on the T2. So we submitted our request along with the justification of lab management. What really helped were letters of support from our pharmacy and our supported services, especially the bone marrow program and the cancer center. The chairman of the cancer center wrote a letter co-signed by the director of the bone marrow program, along with infectious diseases. And that letter, I think, had tremendous influence on us being approved to acquire the instrumentation and to be able to offer both bacterial as well as the Canada. So as I said there at the last bullet, the C-suite has agreed to support our request to acquire the instrument and to test both panels. The implementation, we've had the schedule. We have trained all of our phlebotomists and our nursing personnel in order to promote consistency and uniformity in collection. We've selected and have trained our technical staff on the operation of the T2. And we have 2 people yet to train for our after hours, but that will be done within the next 2 weeks. The other key thing was to create an electronic order, so physicians will be able to order both bacterial as well as the Canada panel. We have put together a guideline in terms of optimal ordering of the test results, and I'll share that with you shortly. We have a go live date of next week, actually, and we're communicating to the services, reminding them who is eligible to order the test. And we're going to call positive results specifically to the ER, to the nursing units, and the pharmacy. Those 3 services will get positive calls. The techs aren't too happy with that, but we're in the business of patient care and influencing patient management and supporting our medical staff. So the emphasis, and I have there in bold, and I can't stress this enough, in your collecting teams, your phlebotomists and nursing personnel really have to emphasize the importance that that T2 sample be accompanied by a blood culture. And in our experience, in our guidelines for ordering, we are recommending in the ER that patients that meet the criteria for the sepsis protocol, the bacterial panel be ordered. For the bone marrow and the solid organ, we recommend the Canada panel be ordered. For intensive care units, medical and surgical, and infectious disease, they can order both. Now, obviously, that's subject to discussion and maintaining flexibility, but we think this is going to help eventually in the appropriate or optimal use of this asset. So I just want to conclude before saying thank you that I would recommend or suggest that the company add Canada aureus to their T2 Canada and possibly consider Canada duvalensis, pneumococcus to the bacterial panel. But in summary, in addition to that, I think it's fair to say we did have a higher mortality and readmission in the control group, as I showed you on that one slide. We asked, do we need 2 instruments at the current time? We don't think so, but that's always subject to change. We also have to realize in explaining some of these data that patients have other comorbidities linked to mortality, not just candemia, for example. Patients placed and were placed on therapy much sooner following T2 reporting. As I mentioned, length of stay probably will not be affected, especially in your transplant and oncology units. We did notice from the discussions with our PharmD colleagues that negative results, negative TT results, do influence termination of antifungal therapy. And if we can identify the agent in 3 to 5 hours before a positive culture, anywhere from 1 to 5 days, I believe that is significant. I do believe that the test should be regarded as an adjunct to blood culture, not purely a diagnostic assay. And between the education and the preparation and the planning and the response to management's financial concerns, we were very successful. It's not been easy, but we were successful in gaining their approval. And we're looking forward to offering the T2 in both the bacterial and the fungal support services for those respective services that we designated. So with that, I thank you very much and wish you a good day.
Hello, everybody, and thank you for continuing on our session. I appreciate the comments from Dr. Snyder on the lab side implementation. My name is Todd McCarty. I am an assistant professor at the University of Alabama at Birmingham in infectious diseases. We've been using the T2 Candida assay for several years now, and I've learned a lot about its process. First, a couple of disclosures to mention. One, I do have a consulting agreement with T2 Biosystems, as well as clinical trial research grants with Snyder and Amplex. I was actually finishing my fellowship in infectious diseases when we first implemented the assay, and so I've been here all along and been heavily involved and engaged in the process. And our original goal was to kind of focus on a high-risk population and using the electronic medical record system and the electronic ordering system to try and help refine and enrich that population. And so with the help of our IT group, we created this checkbox prompt screen. So when somebody wanted to order the T2 Candida assay, they actually got a pop-up with this list of risk factors to click through, and they had to meet some minimum number of risk factors before they were allowed to order the test, because we didn't want a bunch of overuse in low-risk patients that wouldn't really benefit from empiric antifungal usage. What we learned through that process was that we made it a little bit too restrictive, and we noticed this fairly quickly, actually. So our goal was to have something in the neighborhood of a 10 to 20% positive rate. In that first about 1 to 2 months of usage, we were actually running over a 20% positive, and we were missing a lot of desired overlap with other patients in the hospital system that were found to be candidemic by blood cultures. And so what we did was we kept that same risk factor screen. We wanted to keep using that, and it was a valuable source of collecting data as well, and also helping people sort of prompt, is this a test that I want or is it not? And so we just dropped that minimum restriction, kept the screen, left that alone, and just kind of entrusted people to look through it, check the applicable risk factors, and then order the test. The other sort of growing pain that we noticed after the first year or so was a lot of what Dr. Snyder just mentioned, where we really want these done concurrently with traditional blood cultures. We didn't want it ordered on its own. We wanted this to sort of really fall in line with the rest of the workup for anybody's sepsis, whether it's coming through the ER or sepsis that was onset while in the hospital. And so then the order became updated to link the two. So if you ordered a T2, automatically blood cultures were automatically ordered alongside of it. They were able to sort of opt out of sending those if they had been done in like the recent about 12 to 24 hours. And so we didn't want to sort of overwhelm the lab with extra unnecessary work, but we did want to make sure that link was kept. And so we've sort of learned throughout this process, and we've presented data at a variety of meetings over the past few years about how it's implemented and impacted care here at UAB. And so our first couple of abstracts were at ID Week in 2017 with a couple of fellows at that time. And a lot like what Dr. Snyder just shared, definitely people getting a T2 sent were ending up on therapy a lot faster. Most of the time, people were starting in a kind of candidate at the time of ordering the T2. And rather than sort of getting that positive yeast on a positive blood culture gram stain to 1, 2, 3 days down the road. It was interesting, though, in that first sort of look at a year's worth of data that our T2 positive patients had higher average Apache 2 scores and a higher average mortality. The vast majority of the positive blood culture patients didn't have a T2 sent, and so there's an opportunity for education to sort of help make people think of that. You know, I think that's a lot of what drives some of the persistent mortality with candidemia. One is these are sick patients, you know, they're very sick patients to begin with. But at the same time, we just don't always think about Candida. We are so sort of honed in on bacteria and thinking about broad spectrum antibacterials that Candida, unfortunately, I think gets lost in the shuffle a little bit. And then we had this population, a lot like what Dr. Snyder showed and shared, that was identified only by T2 and had blood cultures that were sent, but were found to be negative. And so it's always sort of an interesting thought exercise. You know, if you have this new technology, this new test that is a potential or higher sensitivity than what has been the gold standard in the past, how best do you interpret that? No test is perfect, of course. There's always the potential for false positive, false negatives either way. How do you sort of best define what a false positive is when you have a negative culture, knowing that blood culture sensitivity is a little bit lacking at times? Our other side of this, and I think one of the biggest benefit is the negative predictive value of a T2 and being able to very quickly stop unnecessary antifungals. And so we looked at about 2 years worth of data and we hit that sort of mark that we wanted. We wanted about a 10% positivity rate. We did see an overall increase in our echinocandin usage, which was a little bit sort of contrary to what we expected. We thought we might shorten it. I think there's a balance. While we did show that empiric echinocandin usage was shortened, and this wasn't a part of that actual abstract, you know, it was definitely, people were getting less echinocandins when there was no candidemia or invasive candidiasis. We were diagnosing people that we would have otherwise missed with cultures, and they were getting the typical 2-week treatment course. And so the savings that we had on the early cessation of unnecessary therapy were balanced by the identification of additional patients. I wanted to look at this specifically in an at-risk population. And so we looked at 2 years worth of usage in our solid organ transplant population. How is it being used and being interpreted there? There was a good amount of usage. The bulk of it was in the ICU, which I think is exactly what you would want and expect. We kept that sort of less than a 20% positivity rate. So we're looking often enough that we're not worried about missing a lot of patients. And people definitely trusted the results. You know, the bulk of them were negative. We did shorten empiric treatment with the negative result, and people did not have sort of relapses or issues down the road. And so it was a reliable and trusted test in this patient population that is always a little bit difficult to treat and sort of know which in the broad array of different infections this immunocompromised patient population can acquire and effectively rule one out and stop unnecessary treatment. A couple of years ago at ID Week, we wanted to get a sense of, are there any differences across the different species results on the test? What are the risk factors that most people are popping up with? What is the mortality? And so this was 3 years of data. And 173, we looked at just our positive patients. And all of them had multiple risk factors for disease, most in this sort of 2 to 5 risk factor range, most commonly getting broad spectrum antibiotics and having a central venous catheter in place. Albicans and tropicalis was the most common positive result, which I think is exactly what was expected. But interestingly, while glabrata is our second most common etiology of candidemia based on blood cultures, that wasn't true for this. So there was a lot of parapsilosis that was being picked up. We noticed that, we still had this very high mortality. These are very sick and sort of enriched ill population, and seeing, more risk factors, older age, and then having albicans or tropicalis was definitely an increased odds for mortality. Interestingly, the parapsilosis group, being bigger than expected, also had fewer risk factors for invasive candidiasis and also had fewer risk factors for candidemia. And so that was, I think, an interesting finding, but they still had multiple. So is there some potential for false positives in that group? It's a maybe. I think some of them probably were, but a lot of them were sick patients who you would still want to treat a positive result and make sure that's not something that can contribute to illness in that moment, in that hospitalization. Next up, we wanted to look at, sort of in the same time frame, what was our false negative rate? And so we used a 24-hour window, so 12 hours before a T2 was being sent and 12 hours after a T2 was sent, to better kind of capture, making sure that we're looking at patients that really had the 2 tests sent at them within a close enough time frame of each other to really be able to evaluate them. Most of the false negatives that we did see, and there were about 17, but this was really limited by we're still missing out on doing a good job of linking the 2 tests, the blood cultures and T2. Most of them were species that were detected by the assay. A small portion of them weren't, but we didn't have a huge impact. So a lot of them, there was a little bit of a delay in starting. So they had the negative T2 and it was either not started or the antifungal was interrupted and then resumed once the positive blood culture. But 9 of them, the bulk of the patients, there was no impact on their treatment whatsoever. And so I think it's sort of an indication, A, we have to do better, B, if there's enough risk factors that you're not going to believe the negative result, maybe it's not worthwhile sending the test, just start them on the antifungal. And C, we really have to be better at linking the blood culture tests with the T2 and making sure we can fully evaluate all of these patients and all of these results. And most recently, this past academic year, we wanted to look at eye disease in this patient population. This came up because of a clinical observation. We had a series of patients that were T2 positive with negative blood cultures sent at the same time most frequently, that were then found to have some form of eye disease. And so that was our question, is how often is this happening? Is it similar to blood cultures or is it different? And so we looked at 4 years worth of data. As time goes on, we could accumulate more and more patients. The bulk of our patients do undergo dilated eye exams. Our goal is for all of them. And the minority end up with any sort of eye disease. And so the percentages were fairly similar between the T2 positive group and the positive blood culture group. The bulk of them were asymptomatic and most had chorioretinitis, so not the more invasive vitritis. Most of them did not require any sort of intervention from ophthalmology. But we had the same representation of parapsilosis being overrepresented in the T2 positive group, where it's one that's, we don't really link it to eye disease. We think of the more virulent albicans and tropicalis, and to a lesser extent glabrata, as causing endophthalmitis. Parapsilosis, when you look at a lot of the Candida eye disease literature is a very uncommon finding, but this stands in contrast to that. And so is that something that we've missed over the years? Is there, chorioretinitis with perhaps locis that's been missed and not caught, especially in asymptomatic disease? Lots of questions, I think, still to be asked and answered. A few sort of thoughts from others that I like to sort of show data on, because I think it really lines up with how we've developed and used the test here. And this is a nice paper from Neil Clancy and Hong Nguyen at the University of Pittsburgh, where you really look at where does the test perform best. And this lines up very well with this 10 to 20% desired positivity rate. You want people to be sick enough that there's a real risk, but you don't want them to be so sick that no result is going to convince you otherwise. And so if you look at those positive and negative predictive values, you really see a really high positive predictive value once you go above a 10% prevalence and risk. And you start to see that negative predictive value, while still very high, start to drop off a little bit once your 20% risk of prevalence is met and goes even further. And so it's this narrow band, but a lot of patients kind of fall into this and knowing, is this going to be Candida, or am I going to get fooled into thinking it's Candida when it's actually bacterial in some of these low prevalence populations? And so I think that 10 to 20% is a really good sweet spot. Dr. Arendrup over in Europe has kind of looked at it as a screening tool. And so they did twice weekly T2 screening alongside blood cultures and the Candida mannan antigen in ICU patients that were at high risk, that showed very good sensitivity and a very high negative predictive value. So not just in the moment, okay, somebody has gotten sick and we need to think about a new infection and thinking about blood cultures and sending a T2, but actively screening for and looking for it. Was shown to be a very good and reliable test. I mentioned the other sort of group is, can picking up DNA help discover invasive candidiasis that hasn't led to actual candidemia? And so there is low sensitivity. It is what would be expected, but it's there. So there is this opportunity to diagnose more patients with invasive disease that's not led to the extent of being fungemic. And there is a very high specificity when they're linking them to sterile site or colonizing cultures. And if you combine it with additional antifungal tests, you really have a very good sensitivity and don't lose that specificity. And so I think, it is a great adjunctive test. Like Dr. Snyder said, it can't be used in isolation. It has to be used in connection with all the other things that we normally do to figure out what is causing any person's sepsis. It has to be done alongside traditional blood cultures. It has to be done alongside abscess cultures and sterile site cultures, but can really speed up thinking about Candida and fungal disease. So how do I teach people in our institution about how I want this, how I want them to use it? It's definitely a blood culture equivalent, and starting antifungals in the appropriate time frame for sepsis is always hard. You have to think about it. This is a test that you still have to think about Candida to want to order it. And so better educating people on what are the risk factors for candemia and invasive candidiasis. There is some threshold of signs and symptoms and antibiotics that they should want their patients to meet before sending this test. And once you kind of get into this tipping point where your utility starts to get worse, once you pass above that 20% prevalence, are you really going to believe a negative result, or are you going to think about odd species, different species, and want to wait for cultures before deciding what are you going to do with the kind of candida? If you're not going to believe a negative result, don't waste the resources, don't waste the time. Treat empirically and wait and see what the culture showed. What is still to be learned? So what does usage look like in prolonged neutropenia? In this sort of gut translocation models of neutropenic fever, can you detect Candida DNA that isn't necessarily clinically relevant? how do you better understand this eye disease, and where is this parapsilosis and like linkage to eye disease fit into all of that? I think that's something that's better to be understood. And I think ophthalmologic screening of the retina and in candidemia is something that still people argue and debate about a lot. And there's still a lot to be learned there. Who does need an eye exam versus who doesn't? Is there something different about patients that have a positive T2 Candida and negative blood cultures? We've very much seen differences in those groups here. How much of that is based on provider bias and selection? You know, there's something different about what makes somebody think about ordering this test versus just standard blood cultures. Why is mortality so stable? In spite of all of these things, how has it been so difficult for us to drive that candidemia mortality down? And is there some opportunity to use this test to shorten duration of therapy? It very much feels like a one size fits all. You have candemia, it clears quickly, you get 2 weeks, unless you have some other kind of invasive condition to go with it. But with all of the data coming out in the recent years, looking at shorter antibiotic courses for a lot of common bacterial infections, could this be an opportunity to do something similar with Candida? And can we shorten how long some people need therapy? And with that, I'll finish. I know we're transitioning to Q&A. Here is my email, as well as I'm very active on Twitter. And so if you want to give me a follow there or chat there, I'd be happy to.
So, Dr. McCarty, I have a question for you. Can you please share the impact of the innovative and accurate lab diagnostics you mentioned about T2, your experience with T2 panels, which shorten the time to result in sepsis patients? So in a positive way, why would you use T2 panel with all the detailed data that you shared? So.
Yeah, definitely. So I think, you know, the T2 Candida panel has a very nice niche in that it can be helpful from a positive and a negative standpoint. You know, so when people think about Candida and they want to start antifungals, they can see that confirmation with a positive test in patients with risk factors. But at the same time, you know, I think a lot of the power is in a negative result. The need and ability to perform antifungal stewardship, it always goes easier when you have data and information to give prescribers. You know, it's hard when you have a condition that is, you know, notoriously hard to diagnose, which I think most fungal infections are. You can come back and say, okay, the test is negative. You really don't need this extra medicine. It's okay, it's safe to stop, and especially as you identify different diagnoses.
Thank you. Thank you. Thank you so much. Dr. Snyder, I think the participants wanted to hear the same viewpoint from you about the impact of a lab diagnostic test, which shortens the time to result in specifically in identifying sepsis causing pathogens in these critical patients.
Well, in addition to what Dr. McCarty responded to, I firmly believe with rapid diagnostics, and in this case, the T2, if an answer is available in 3 to 5 hours, and I will also say this is something we're sharing with our group, if we're running a full capacity assay, 6 specimens at one time, which may happen, that's going to increase the time of testing to the point that it could go up to 8 or 9 hours. But the purpose is to get an answer that is reliable, but most quickly as possible, for the physician to make a decision to get them on appropriate therapy sooner or not to treat. Those are all clinical correlations. My job is to make the information available to them and to allow them to determine their management plan based upon 4 hours from now versus 2 days from now. So it's our responsibility to release those results as soon as possible, knowing that they are accurate, but being available to respond to questions as to certainty Are you sure it's positive? And then explain to them a little bit how the system works. I don't mean to get detailed, but the T2 system is not based upon nucleic acid. It detects whole cells. And we have gotten physicians used to PCR, nucleic acid amplification, but this is not a nucleic acid-based test. But it gets back to the fact, if I can get the results sooner rather than later, that gives the physician an opportunity to better plan his or her management and selection of therapy, and hopefully more targeted therapy. So we're honoring our responsibility as a laboratory.
Thank you. Thank you so much. We have a question for you, Dr. McCarty. As per your experience of past few years that you've been using the T2 panels at your facility, could you share the trend of the test utilization for T2 panels at your facility, please?
Yeah, definitely. So usage has gone up every year. I don't know the exact numbers of how many we used last year, but each of the prior years that we've looked at data, the number goes up. I think as people get comfortable with the test and how to use it, I think it also is a familiarity thing. You know, they remember that the test exists, or they hear about it from other people, and they're more sort of willing and interested in sending the test to begin with. So usage has definitely risen every single year. And I think that's good. You know, I think to Dr. Snyder's point, we all want data. We want as much data as possible, as fast as possible. And I think it's been a valuable addition to how we manage patients here.
Yes, thank you. And I know clinicians always need data, and especially for something as critical as sepsis, this is really very valuable. Thank you so much, Dr. McCarty. Dr. Snyder, this is a question for you. What are the factors you and your colleagues took into consideration while choosing the patient population to test? I know you did share a lot on that, but just maybe.
Well, once again, to reemphasize, we asked the question, what patient populations account for the highest incidence of candemia and bacteremia? And as indicated, the patients populations that we selected are those, the ICUs, the oncology, immunocompromised from transplants, solid organ, bone marrow, and so forth. We also did have a question when we were meeting with management. We had some physicians there. It was a multidisciplinary team. And the one physician said, well, I'm not in any of those services and I want the test. How can I order it? And so to kind of respond to Dr. McCarty's a little restriction, we lifted the restriction, said, yes, you can order the test provided that infectious disease is consulted and approves the test. So anybody can get it, but you have to get an ID approval before it actually is submitted. We also had to consider that 24/7. These technologies are designed to run 24/7. And it's our responsibility to ensure that we have the support personnel from the technical perspective in terms of staff to do this. We've been lucky to be able to convince administration the importance of this. That was taken into consideration as well. And as I said, these tests are designed to be run or generated 24/7. I really believe it's a disservice to the medical staff and patients not to maximize their utilization in such a manner. So select your patient population and at the same time, realize who you're serving and the purpose of a laboratory.
Thank you so much, both our key panelists. And I do want to share with all the participants that all the speakers would be present at the T2 virtual booth on June 21st between 1:30 to 2 p.m. EST to answer any further questions. Thank you so much.